Hi, my name is Catherine Herman, and today I'm here with Lena Smirnova and Thomas Hartung at the Center for Alternatives to Animal Testing. And we will hear a little bit about the history of the center, about the current projects, the programs we are running here, and also future directions. So CAAT's history is quite interesting because it's one of the oldest 3R centers. It was founded in 1981. Thomas, can you tell us a little bit more how this happened? What was the idea? What were the first projects of CAAT?
Yeah, sure. I mean, I was at the time not here. I was finishing high school in Germany. It was in the late 70s and early 80s that the cosmetic industry actually came under pressures. Henry Spira is one of the very well-known activists. There's movie films about him, and he did actually do these one-page ads in the New York Times. How many rabbits did Revlon blind for beauty's sake? And this had an impact. And it was the cosmetic industry which said, we have to find out whether we can really credibly employ the science to replace things like the infamous rabbit test on the Drace test on rabbit eyes.
Yeah.
So they came up with $1 million, which was a ton of money at the time, and found here in the School of Public Health with Alan Goldberg, my predecessor, somebody to set up the center.
And when did you start working or taking over CAAT, and what did you change then? Like, what was the state you took it over at, and what was to follow in the next over a decade? Right?
Yeah, I mean, in the 27 years Alan did lead this center, it had become really a very reputed source of scientific information. So really very different to what animal welfare activism is producing. It's not propaganda, it is not trying to move the hearts, but it's trying to move the science. And Alan really made it an information hub. So I joined the center first in the scientific advisory board when I was still head of the validation body in Europe for the European Commission. So I knew since 2003 what the center was doing very well. And when they approached me in 2008, I said this could be a cool thing to do, to be in an environment of international public health, have the possibility with the branding of Hopkins to promote alternatives to animal testing. So in 2009 I joined, and now 15 years later I'm still here.
That is most wonderful. What I also recall is that my predecessor here at CAAT, Joanne Solo, was quite there from the start, or quite early on, and she showed me all her materials she had, and they did quite a lot for school children, informing them about the 3Rs concept, the 3Rs principles, what they stand for, and you recently picked that up again. You started educating younger children, like 12 or 12 onwards. Can you say a little bit more about your activities in this area?
Yeah, I think being an information hub is really about informing all stakeholder groups, and the stakeholders of the future are the kids. So to the extent we find resources, we are addressing this. We have most recently teamed up with Frontiers, the publisher. They have a very interesting project which is called Frontiers for Young Minds, which is targeting children from 8 to 15. So they are translating scientific articles into kid amenable versions, or have them written. And last year, together with the Swiss 3R Center and some other partners, we developed 3 articles on the 3 Rs of alternative methods. I wrote the one on replacing animal tests, there was one on refining animal tests, and there was one on the reduction of animal numbers. And we're continuing this, so we are at the moment working on the 5th of these articles, because we believe that this is an investment by attracting to this area of work people who are up to decide what they might be studying in the future.
Yeah, that's a good point. I read that they are also reviewing the articles. And this is really amazing, because then you actually need to explain it in an understandable way. And I think that's something that scientists sometimes forget, that there are people who might not know everything already. And so I found this quite interesting. And also what they did, for example, when they explained organs on chips, and they did this kind of with a Lego style, that was another thing. So yeah, I totally agree with you. We need to cater more to kids as well, not only to students and early career scientists. However, early career scientists obviously is another big thing that you are focusing on. You are helping early career scientists. Can you maybe say a little bit more about what we have in terms of funding programs and your teaching you're doing?
I mean, first of all, a scientific career typically starts already in school. You develop an interest in going into science, technology, engineering, or medicine, what we call STEM now. But the specific spin to look into animal test alternatives is something which typically develops later. But at each and every step, you can start influencing this. Materials which are made for kids are seeding something. It is the young career scientists who have to be attracted to making clear to them that this is the future. Because, to be honest, 100 years ago, when most of medical research did really start and take off, it was the only to go to use animals, because there was no cell culture, there was no computers which we could use as the main alternative technologies. And this has imprinted on many with the idea you cannot write a new line in a textbook without a new knockout mouse which has been produced. I believe that with these disruptive technologies, which at the moment are changing life sciences, AI, artificial intelligence, the lab on chip type of approaches, organ on chip type of approaches, this is really the time where we can drop something which is 100-year-old technology and replace it by what is really giving us human relevant results faster and cheaper.
Mm-hmm. So what do you think, what do you have planned for the next years of CAAT? Do you have a vision already what you want to achieve in the next decade?
I think that the big change is that it is no longer about trying to find alternatives. I believe that we really have most technologies available. It's about fine-tuning, getting them a bit better, but the main thing is actual change management. Mm-hmm. It is about not doing it because we have always been doing it like this. It is about not requesting an animal test as the final proof in a scientific paper for something which has been elaborated with much more advanced technologies in the paper. Yeah. So this is something where a change in mindset is necessary. And the most important part of this is for me initiating doubt. Showing my colleagues that we are overestimating the value of animal tests. Showing them the problems in reproducibility. Showing them the costs of these. Showing them that societal acceptance is not with them. And all of these are driving them at least as much as ethical considerations. You know, we are a 3R center. I think we should first of all explain what 3Rs are about.
So the 3Rs of animal experimentation are replacement, reduction, and refinement. So we want to first of all, try to not use animals, but more human-relevant methods. The second is to slowly reduce animal experiment. And the third, that is my main focus, is refinement. And that stands for doing anything you can to reduce the suffering of the animals who are in the experiments.
Yeah, exactly. So when you really have to do an animal experiment because there is no replacement, refine it. But what does this mean? How can you make this a more pleasant experience for animals?
There are different areas, of course, or you can say what is part of the life of the animal. Obviously, a huge part of the animal's life is the housing or the environment in which the animal lives. So you can do improvements of the housing and also the caretaking of the animals, ways of handling. For example, a standard thing that unfortunately still often happens is that they pick up mice on their tails, and that's really distressing for the animals. And they have implemented, or they should have implemented by now, the tunnel handling, where they can walk into a tunnel and then they are picked up. Or then also the other thing is cupping them so they don't feel so scared. That's just an example. Other things in other areas for refinement is experimental refinement. That means anything you can do to make the experiment less averse for the animal. And that, of course, also includes some standard veterinary care, I would call it, because a good analgesic protocol or anesthetic protocol is something that should be standard practice for any veterinarian, but it's still something we have to focus on in the laboratory animal setting, because not everybody who's doing animal experiments is a veterinarian. Okay. And then, of course, there are other areas, like one big thing is the so-called humane endpoints. That those are endpoints when they are reached, you know that the animal is suffering to a certain extent, and that should not be the case, and then the animal normally is killed, or they are taken out of the experiment to recover. But it's a threshold that shouldn't be overstepped, because there's something called a harm-benefit analysis that you do to weigh the harms of the experiments against the benefit for humans. And the rule is that the harms for the animals shouldn't exceed the benefits. And of course, if you then exceed a certain pain threshold, that makes the harm side exceed. Yeah. So all those areas I'm teaching at Hopkins, and also in another open webinar series that CAAT is doing. Lena, I'm very interested in knowing more what you're working on at the moment with the MPS program. I know that, of course, CAAT is organizing this World Summit, and that's super exciting. But I would also love to know more about your lab work at the moment.
Sure. First of all, what is MPS? So people know, MPS is microphysiological systems. And microphysiological system means that it's new generation of cell cultures which are developed over the last decades to advance the in vitro safety and efficacy testing of drugs, but also safety testing of chemicals without using animals. And what is important about MPS or microphysiological systems, they closely recapitulate our organs and organ functionality. So they kind of mimic the architecture of the organ and functionality of the organ, which is ultimate goal for our replacement models, to have it closer to in vivo situation. And there's a huge need in the society to bring all those people who are developing this type of microphysiological systems or MPS together, so that they can exchange and promote their technologies, but also to make sure that developers are talking to regulators are talking to end users, and there is a loop is closed. And we organizing, as you said, World Summits on that. So CAAT is heavily involved because we received a grant from NCAT to initiate this process toward International MPS Society, which we are funding right now in the moment. And we can see already how field is growing, because we had in the first summit, we had 450 attendees in New Orleans. Now in 2 months, we will have that in Berlin. And we have already over 1,000 registrants for that. So it's a huge increase in success in that sense. So from the research perspective, how we contribute from our research, from our lab activities for MPS field is mainly focusing on the models of the brain. So we are developing models from induced pluripotent stem cells, so the stem cells which can be reprogrammed from any type of cells from your body, so everybody can donate a little bit of skin, a little bit of blood, and we can reprogram them into the stem cells again, and those we differentiate in different types of brain cells in a 3 dimension, right? So they are forming these connections called synapses, they interact to each other in 3D, which is again closely recapitulate the functionality of the organ.
And I recall that at the beginning of the COVID-19 epidemic, you actually did tests on the so-called mini brains, and were one among the first to show that unfortunately this virus also affects our brain. What else have you been doing with this
that's
model?
right. COVID was intense time to research. But my particular focus is neurodevelopmental disorders, in particular autism. So recently CDC released the new numbers for 2023. We have 1 in 36 children diagnosed with autism in the US, which is the huge number, and it's continuously increasing if you look back in a couple of years. So in my research, I'm focusing on understanding what contributes to that increase. So we cannot explain it only by the genetics or only by, let's say, better diagnostic or awareness, right? So our hypothesis is that interplay between genetics and certain environment, which can come together, interplay, and then trigger the disease or put people on different spectrum of the disease. Like severity can be different. And this is what we are addressing with our brain organoids model, so with our iPS-derived model. And we have iPSCs with different autistic genetic background from patients or modified cells with a CRISPR-Cas9, and then we apply different environments to that to see how they respond.
And then you look at different toxicants probably,
exactly,
to see what that does. So can you maybe explain a little bit for the listeners who are not familiar with why we're not using animal models at CAAT? What can you say for the example of autism? Why that is not the way to go?
There are many aspects to that. Of course, ethics is a major one of big ones, but it's not only about ethics. And there is a strong push from also general public to replace animals for testing. This is the ethical concerns. But also, we do see that the problems is that the animals not necessarily recapitulate the human physiology as we wish they would. And very often animal tests end up not be predictive for humans. And this, especially with these neurological conditions, if you look at clinical trials, in general, over 90% of clinical trials are failing after really successful preclinical animal trials, right? So
is there autism in other animals, or is that a very specific human disease?
I mean, there are models, behavioral models of animals of autism, but certain genes, for example, are not exactly the same as humans. And there's another factor as idiopathic autism, for example, which you cannot model an animal. So animal is only, you can introduce one mutation, for example. We are currently working, for example, with Kennedy Krieger. It's not autism, but it's rare disease, leukodystrophy. And they're developing Kennedy Krieger actually also and CHOP, and they're developing therapies against these diseases. And for example, they approached us because their animal model is not working because this gene doesn't exist in animals. So they cannot prove the therapy, anti-gene therapy, with these animals. So they approached us and asked for help with our model.
Another new program of CAAT is an initiative that we are doing in collaboration with another lab here at Johns Hopkins. It's about organoid intelligence, and it's focusing on Alzheimer's disease. Thomas, can you explain a little bit more about this initiative and what your goals are?
Sure. You will already have heard from Lena about our work on brain organoids. So this is a technology which tries to reproduce tiny brain equivalents. And these are ultimately starting to develop functions of a brain. So what is the function of the brain? It is cognition, it is memory, learning, what makes us intelligent. And actually, these little systems, there's increasing evidence, are capable of doing exactly this. So organoid intelligence, or OI, is the combination of these organoids with artificial intelligence. This is why OI, organoid intelligence. So we have teamed up with about 80 researchers in the meantime. We have had a community forming workshop and just published the concept of organoid intelligence as a new discipline. And this is an infectious idea. It is really opening up for brain diseases to give us an endpoint which is really meaningful. And you have to imagine 25% of all clinical trials are done on brain diseases. So it's not only Alzheimer, there's neurodevelopmental diseases, there is multiple sclerosis, there is Parkinson, many other things, and all of these can benefit from models where we have sensitive endpoints which are really reflecting brain function. And this work is sponsored by Hopkins so far. We were the lucky recipients of some major funding from Hopkins, and we work with several partners in biomedical engineering, with the applied physics laboratories and others, to really form a core, an epicenter for developing this in a global initiative. So our most recent release of this concept led to about 600 press hits. So there's tons of people interested joining now this community. And the latest news is that the National Science Foundation is now going to invest into an engineering organ intelligence program. Even the White House has made this part of their bold biotechnology goals last month and said we have to develop organoids which show these capabilities.
Another question I have is about green toxicology. What is that, and what has CAAT been doing in this area?
Green toxicology is a part of green chemistry. Green chemistry at the moment is very much in the news as part of the sustainability goals, making chemical industry on the one hand more energy efficient, but also reducing the risk of chemicals. So 2 of the 7 principles of green chemistry are actually toxicological principles. One is, for example, benign by design. So let the chemist already create chemicals which are less toxic. And this fits extremely well with what we are doing, because first of all, why develop something for tens or hundreds of millions for a drug, 2.4 billion, before you really bring it to the market? If you can already early on anticipate the effects. This is something you cannot do with animal tests easily. There's also an acceleration of the process. Time to market is extremely important for industries. To give you one example, if you want to know whether a new chemical is carcinogenic, the animal experiment to do so does cost you almost $1 million. You have to wait for 5 years until you have the results ready, and you need about 10 kilograms of this substance in order to run the tests. Tell a chemist to produce 10 kilograms of something which is not yet in production. So it is extremely important to have methods, ideally computer-based methods, which tell the chemist this substance is likely to produce cancer, better drop it and put your money and your time on something which is having a much better profile. And that's the basic idea of green toxicology. So it is using the very same things which we use to replace the regulatory type of testing, but say we do it so cheap and we do it so fast, you can anticipate this. In pharmaceutical industry we call this front loading of toxicology, and this saves tons of animals and makes those which are finally used also suffer much less because they're not exposed to real toxic stuff.
I was surprised because our colleague Alex Mertens published the first textbook on this. So it shows that it's still a rather new field, even so it's kind of, as you say, it makes so much sense and it's so important.
Yeah, we started this in 2014. We worked with colleagues in the EPA and in the chemical industry to develop this idea. And Alex has been part of this from the beginning. At the time she was still a PhD student, but she had this interest and organized our first meetings and the first papers, and that's how it all started. And in the meantime, this has been really highly recognized. For example, I'm member of Apple's Green Chemistry Board. Yeah, this is just 7 people, external to the company. To the best of my knowledge, the only external advisory group Apple is having at the moment.
Oh, that's interesting. And can you give this example? A few years ago, you ran this huge data set, and you actually kind of found out how much better it is what you were doing, because the animals that we usually use were not very predictive. Can you say a few words about that?
Yeah, You know, there's at the moment one of the most disruptive technologies in our entire life is artificial intelligence, machine learning. And you can do machine learning when you have data. So what we decided in 2016 already was to make the largest databases machine readable. So we really enabled a computer to read the submissions to the European Chemical Agency, and this work led to a database where we had 10,000 chemicals which were tested for each and every endpoint by on average 3 times. Incredible, billions of dollars of testing costs which went into this, and these are high quality data, all of them OECD test guideline studies done under quality assurance schemes, and this database at the time was the largest toxicological database in the world. And then we used this on the one hand to show how little reproducible animal tests are. Can you imagine that for the 6 most used animal tests, if you run them 3 times, you will find a toxic property only 2 times of the 3 experiments. And this means already we should be wide open for finding something new. But we showed then in 2018 that we can actually do this machine learning. And we clearly outperformed for 9 OECD guideline tests, which use about 60% of all animals in toxicology, that we outperform the animal test with respect to its predictivity. And you press a button and you have the answer in seconds.
Another successful program of CAAT is the evidence-based toxicology collaboration that commenced in 2011. Thomas, can you tell me what your ideas were? Why did you come up with this collaboration, and where is it heading today?
Actually, I would date it back a bit earlier. I was working as a pharmacologist in infectious diseases. So I was working also clinically with patients, and I was witnessing a revolution in clinical medicine, which is called evidence-based medicine. They understood at some point there's far too much information. Nobody can read it anymore. We need somehow to condense to what is really true and find ways what is the evidence and what is the noise. Yeah. And when I joined fully the field of alternative methods in 2002, I had the idea that I could translate evidence-based medicine to toxicology, which became the main topic already working in the European Commission. So I started this field by also organizing the first conference in 2007 in Italy, where we had 170 people from all continents and formulated a declaration of COMO towards evidence-based toxicology. When I then got an offer here in Hopkins for an endowed chair, we found the name of evidence-based toxicology as the denomination for my professorship here and chair. And so we did shortly after initiate the evidence-based toxicology collaboration. So it's all about showing what is true, applying very rigorous approaches very objectively in order to assess current and new methods in order to move forward. So I had a lot classical animal testing in mind and the new methods and how to compare them in a way that nobody can doubt it, can tell me, you're biased, it is just ideology. I wanted to have the tools which are waterproof against such type of criticism. But there's others who had at the same time saw more and more information becoming available, which is often contradictory. So you want to know about a chemical, let's say formaldehyde, and you want to know whether formaldehyde is producing cancer or not, or bisphenol A in baby bottles. There is now, as far as I recall, 12,000 scientific articles about bisphenol A, and still there's a controversy what it does mean. And this is why we need ways of handling the information better. And this is what the evidence-based toxicology collaboration is about. And we were joined by representatives of the Food and Drug Administration, of the Environmental Protection Agency, the National Toxicology Program, but also the European Food Safety Authority, for example. And over the first decade, with the Board of Trustees, we have developed principles how to do evidence-based medicine in this field. And just about now, we are moving this to a membership-driven organization. We created a journal for evidence-based toxicology, and we see that essentially from no papers which were done like this, systematic reviews is what the main tool is. We are now seeing more than 1,000 of systematic reviews per year being published in our field.
Mm-hmm. And can you explain, I remember the first project was a very extensive one on the zebrafish. Can you explain what was done there and maybe a little bit more about systematic reviews, what that tool is and how it's done?
A systematic review is very different to the type of reviews we write in the scientific literature. It is not just I tell you what I think about a certain area and I cite whatever I like, but it is about a very defined question. So I'm saying I want to answer this question. Then next I describe or develop a strategy. How do I find the information? And what will I include? What will I exclude? So let's say I want to know, let's say bisphenol A. I want to accept studies which have a control group, which have at least a concentration response and whatever. They need to be written in English and they need to have proper statistics. And these are the criteria which I define before I execute my work. And then I need to find a way of combining them. We call this meta-analysis. So how do I combine the papers which in the end are left over? And this is the evidence-based approach. There's some more qualities, evaluations and so on, risk of bias analysis. But it is a very, very accepted approach to handling diverse and incomplete information sets.
Mm-hmm. And wasn't one of the big things when the systematic review started, I mean, they first were pretty common, obviously, for clinical, in the clinical setting, but not so much for animal research. But wasn't one of the big things that we saw how poor the quality of many studies was? And can be like that we couldn't even include everything because of the poor quality of some of the papers?
This is the big lesson you get from this approach. When you define criteria, what you are willing to accept as evidence, you start, needs to have proper statistics. Turns out that 1/3 of scientific publication does not have proper statistics. Needs to have a control group. You would think, that's the minimum, but you cannot imagine how many studies are not done with proper control group. And then if you go further, you have to be done the experiment blinded, so that you don't know which animal was treated how. So it is really a very important feedback to science. What is admissible evidence and what is not. So clinical practice has changed. The quality of clinical trials has changed because of evidence-based medicine. And I would like to see the very same thing for the animal studies which are done, as well as for the in vitro work, because in the essence it's all the same. Document it properly, have a study design which is robust, and be able to convert it in a way that you can combine studies to create an overall conclusion.
Yeah. And that is where our good cell culture practice comes in. That's another thing that CAAT has been driving. Can you explain how you got into this and why it is, of course, so important? You already said a little bit about this. But.
I mean, the three things which count in science is quality, quality, and quality. So an experiment which is not done with quality is an experiment not done, it is toying around, it is a waste of money. Experiment which is not reported in high quality, so that nobody can repeat it, is just the same. Even if you did the best possible science, if nobody else understands it and can repeat it, it is worthless. And so it is extremely important to develop quality standards. Such standards are around only for very few areas, mainly where regulators are involved, good laboratory practices. So in 1996, I organized the first symposium on this topic, good cell culture practice, at the time still in Germany, and then moved this to guidance which we published when I was working for the European Commission in 2005. 2005, however, was 1 year before these wonderful human stem cells were developed, which now allow us to create these advanced cell cultures. And so it was about time to develop an extension of this. We call it Good Cell Culture Practice 2.0, which we published last year, which with the input of more than 100 people in 8 years' work, resulted in now something which is covering also the advanced cell cultures of today.
Yeah, what I found interesting when we think about HeLa cells, for example, how often there was contaminations and we didn't check for that. That's just one thing that should be standard, right?
There was an interesting study most recently in China where 400 random cell lines were checked, and of these 400, almost 100 were not what they were supposed to be. They were often from other species, of other organs, and this is not to single out China in any way, because we have older studies from essentially every country where we looked at where exactly the same has happened. And we also have to assume that 20 to 25% of the cell cultures in academia are infected with mycoplasma. And this is a terrible change to the cell being infected with these pathogens. And very often, despite the fact that it is easy to test, we don't control enough. Yeah?
Thank you so much for your time today. It's been a very interesting conversation.